No, the next talk. It's the next session.
The next session.
Well, maybe we can do in the during
because
the
we
in the
can
in
start.
the
Oh,
in the
sure,
rec.
yeah.
No problem. Is this yours?
Yeah.
Okay. I just take it, and I will remind me after the keynote, the lunch break, we put your presentation
Okay,
inside.
sure.
Thank you. Okay, let's continue with the first session that we improvise a bit. Now, I promise you that we will follow the schedule. You're the first. Yeah. Okay, now is the time for the flush presentation. So the first one is Abi Ispiritaba with the title Allevin Efficiency Estimate Accurate Gene Abundances from DSC RNA Sequence Data. Thank you.
Sorry. You might have to drag it out. Sorry. Oh boy.
You can follow your presentation there.
Thank you.
Remember, you have
Yeah.
3 minutes. Oh,
Fine.
five
Okay.
minutes.
Yeah, I got it. Hello, everybody. I'd like to start by thanking to invite me here, and I came all the way from New York, and it's been hell of a ride. Okay, so I work with Rob Petro, and we mainly work on quantification. We worked on RNA-seq quantification and single-cell quantification. And today, the idea is to talk about Elvin, and we design a tool which can be used for single-cell RNA-seq quantification. And if you were earlier in the talk, Barbara talked a lot about, and it was a very good talk about how droplet and microfluid-based signal-cell technologies are changing our perspective of how you are analyzing the gene expression data. So one of the biggest shortcomings of the current analysis or all the industrial protocol is that you're going to ignore a significant fraction of the read. If read, like I mean as a read, small reads which are generated generally in FASTQ files. So if the read is gene multimap, you're going to just toss it away. And especially if you're talking about single cell, it's costly, and it can be as big as 23% of the data. So basically, 1/4 approximately, you are just tossing it away. So what can we do more to really use them and make better gene expression estimates to predict the better gene expression estimate? So what we came up with is this. It's very intense. I'm not going to follow everything from top to down, but it's end-to-end model. It starts from FASTQ, and it models all cellular barcode UMI-level deduplication, and at the end, it gives you gene-level estimates, which can be used by Surat, Monocle, and all the other downstream tools. It has been published in Genome Biology recently, and we are very excited about it, and it's changing the field in terms of being the state of the art. So just to give you a slight idea, I'm going to be presenting the poster in the afternoon after this talk, I guess. But we are directly competing with CellRanger with this 10x based tool, and we are magnitude. If you look at the left extreme, we are magnitude time faster. We use less memory, and we are more accurate too. So again, if you want to know more, come to the poster or look at the paper, and we are supporting it through multiple our tutorial website. And thanks for having me and listening.
Thank you. Thank you, Arvin. The next, please. Okay, the next is Michael with the title of Comprehensive Pipeline for Processing, Deconvolution, and Visualization of Complete DNA Methylation Data. Thank you, Michael. That's perfect, sir.
Hi, I'm Michael. I'm a PhD student at Saarland University, working mainly in the field of computational epigenetics, more specifically on DNA methylation data. So I'd like to present very briefly to you a very collaborative project with people from the DKFZ in Heidelberg, but also from the LIH in Luxembourg. And it deals with reference-free deconvolution of DNA methylation data. So first, let me briefly introduce what is deconvolution. So imagine you have a biological sample, let's say a blood sample. This sample is bound to consist of different cell types. And the task of deconvolution now is to estimate the proportions of the different cell types in a sample. And if you now have the reference profiles available and you furthermore know which cell types to expect in your sample, it is rather straightforward to also infer the proportions. And many methods have been proposed to perform this reference-based deconvolution. But we here focus on a method for reference-free deconvolution, meaning that we don't have any a priori assumptions. So we don't assume which cell types to expect in our sample, and thus both estimate the reference profiles and also the proportions. And a while ago, we proposed a method to perform that, which is called MEDICOM. And this takes as input a DNA methylation data matrix with CpGs in the rows and samples in the columns, and then performs a decomposition into two new matrices, the T matrix, which are these estimated reference profiles, and the proportions matrix. So you will note that we here on the left-hand side do not call it estimated reference profiles, but we call it LMCs, latent methylation components, because later we found out that often these columns of this matrix do not represent cell types, but something completely different. So this is a very nice approach, but it has two problems. So data needs to be extremely clean, so we need some thorough processing. And furthermore, interpretation is rather challenging in comparison to the reference-based method. So we therefore proposed a complete pipeline to do all of this basically in one go. And when we then applied this pipeline to a lung adenocarcinoma dataset, to a solid cancer dataset, we first found out that after we accounted for potential confounding factors using independent component analysis, we do not distort the overall distribution. And later on, when we then correlated the two matrices to certain things, we found out that we have one component that is significantly linked to the immune cell content of the sample, potentially indicating tumor-infiltrating immune cells. So just very briefly, a summary, we here propose a method or a pipeline to perform reference-based deconvolution of complex DNA methylation data. And with applying this framework, we found out that we found indications of tumor-infiltrating immune cells in solid cancer samples. So if you're interested more into this topic, please come to my poster at number 16. Thank you.
Okay, thank you, Michael. The next flash talk is from Melissa. And the title is Structure-Based Drug Repositioning Reveals a Well-Known Cancer Drug as B-cell Activation Inhibitor. Melissa. Thank you.
So, hi everyone. I'm Elisa Dasme, and I'm a PhD student at the Biotechnological Center of the TU Dresden, Germany. So this project was actually a collaboration with Daniel Parisi from KU Leuven, Belgium. And with this project, we proved the potential of structure-based drug repositioning, which was actually able to uncover this cancer drug as a B-cell inactivator. So the motivation of this project was actually autoimmune disease. So we know that there are severe disease where actually your autoimmune system attacks your body. There are some treatments available targeting T cells, which are the ones mediating this autoimmune disease, but these treatments have severe side effects, and also they are mostly cytotoxic drugs, meaning that they kill the cell. So you don't want to get rid of your autoimmune system, right? So this is not good. But recent studies have shown that actually B cells are also involved in autoimmune disease, and they are able to reactivate T cells after their disactivation. So we said, okay, let's try to deactivate B cells instead. And the first step we did was identify targets involved in the B cell activation or inactivation. And this was done by people in Leuven. They ran an RNA screening in the full human genome, and they identified 22 kinases actually related with the activation of B cells. So the first question we said, okay, is there any available inhibitor already for these kinases? And the answer was yes. We identified 22 inhibitors for these kinases, at least one of these kinases. And actually, one of them was already an autoimmune treatment, tofacitinib. But the rest of them were mostly cancer drugs, cytotoxic drugs. And again, we don't want to kill the cell. So we said, okay, let's try to find something else. And for this, we applied our structure-based drug repositioning. So our screening is based in the similarity of the non-covalent interactions between drugs and proteins. And to perform the screening, we encode this pattern of the interactions with the specific angles and specific distances in the binary fingerprints, so containing 1,155 patterns about these pairings of interactions. So what we did is we took all available structures of the kinases in PDV, we generate the fingerprint defining the profile of the interactions, and we run a screening over the full PDV, trying to get drugs with a similar binding mode, being potential inhibitors of our kinases. And we identified 22 new inhibitors that they were actually validated with a kinase inhibition study. And now you see that only a few are cancer drugs. We have also infection drugs or treatments for infection, and also some diverse other indications like Alzheimer, Parkinson, and so on. So we extend the scope of the indication in these drugs. So we wanted to go even further, and we did a dose response study, and actually ibrutinib and suramine, 2 out of these 20 drugs, were micromolar inhibitors of the same target, BEGFR2, which is one of the kinases that we identified before. So at the end, we had these 3 potential deactivators of B cells, ibrutinib, tofacitinib, and suranine, and the guys in Leuven did a B cell inactivation test based in 2 indexes. The first one was measuring whether the drug is able to inactivate B cells without killing the cell, and the second one, the specificity index, is checking whether the drug is able to inactivate only B cells, not T cells. And finally, only ibrutinib was positive in both parameters, and this is how we came up with our results. So the repositioning is explained with the interactions, similar in both the target template and the drug template. And the cool thing is that the drugs are different, the targets are different, the binding site residues are completely different, and also the organisms. The only thing that is shared between the 2 complexes are the interactions, and we were able to find it. So if you want to know more about this, you can check our poster, number 5, or tomorrow you are all invited to the 3D COSIG. I'm giving the full talk at 3:40. So thanks.
Okay, thank you, Melissa. The next flash talk is Noorram Fadra. The title is Characterizing Allele-Specific Expression Using Whole Exome and Transcriptome Sequencing to Improve the Diagnosis of Rare Genetic Disease. Thank you, Noorram. Let's try this work.
Good morning, everyone. Thank you, Alex, for the introduction. My topic deals with a pilot approach for categorizing allele-specific expression associated with loss of heterozygosity using whole exome and transcriptome sequencing for improving the diagnosis of rare genetic diseases. So first of all, a brief introduction to rare diseases. They are defined as diseases that affect a small percentage of population. For your knowledge, about 1 in 2,000 in EU and about 1 in 200,000 in the United States. But even though they collectively affect, they are said to affect only a small percentage of the population, the over 8,000 or so rare diseases affect 10% of the world population, which is about 350 million. To give you an idea, 6 to 8 million in Europe and about 30 million in the United States. Unfortunately, 50% of those affected are kids, mostly infants who die within the first year of birth, and about 80% of these rare diseases are caused by single gene defects. Currently, because of the boom in rapid DNA and RNA sequencing, particularly whole exome sequencing is being used in order to solve these cases, but unfortunately, the diagnostic rate of whole exome has plateaued over the past 5 years, which has pushed us to sort of explore the utility of transcriptome sequencing in this scenario. And one of the phenomena that transcriptome sequencing is particularly being used for is for evaluating allele-specific expression, which refers to variable or unequal allelic imbalance in a given transcript. It is associated with rare diseases in cases of congenital heart disease, Prader-Willi syndrome, and the Angelman syndrome. To sort of draw your attention to the factors explaining the association of loss of heterozygosity with ASC. The top panel is all of what our genome would look like in the DNA, and the bottom panel is the instances that would happen in case of RNA. So the figure to the farthest left would explain normal DNA and normal RNA. The second figure explains in case of abnormal promoter methylation, wherein you would lose one allele and see expression of only the second allele. And in the third case, if there is a truncating or deleterious variant in one of the alleles, the cellular machinery actually rescues the normal by completely deleting the defective allele, and so you see expression of just one transcript over the other. In order to identify that, we came up with a very preliminary approach that just goes by filtering the DNA VCF for very high-quality variants and picking up heterozygous mutations that are then mined into an RNA variant calling file, which are homozygous. So you mine all the DNA heterozygous variants and mine those corresponding variants in the RNA file for anything that is homozygous. On the right, the figure you see is we obtain basically 40 to 60 variants on average in every sample that actually show this behavior. But then we also noticed a problem that most of these mutations, even though they are genotyped as homozygous in the RNA, their frequencies show otherwise. And the reasons for this could be many. It could be mapping biases. It could be differences in many variant callers. And so there is no effective way of actually evaluating and characterizing allele-specific expression in rare diseases. To combat that, we came up with a workflow that is based on computing the allele frequency using raw data simply by using the alignment files instead of using complex variant callers. When we did that, and I won't go into the technical details of this, but you can come to poster number 27, we realized that we actually have a huge set of calls that overlap with the VCF-based method of measuring allele frequencies, but this gives us a larger umbrella of mining variants that actually show a homozygous behavior in the RNA given their heterogeneous makeup in DNA. And finally, once we have those variants in place, it's easier for us to mine genes that actually show the loss of heterozygous behavior and then mine those genes in order to understand why the LOH behavior is being seen in the first place. There are some reasons that I explained, including abnormal imprinting and nonsense-mediated decay, that could explain loss of heterozygosity in those genes. But there are a small subset of genes which will show allele-specific expression that cannot be explained by common factors. And so the binning of genes in this way actually helps us understand why some genes would show unexplained loss of heterozygosity. With that, I would like to conclude that we implemented a pilot approach for identification of genes showing allele-specific expression associated with LOH in a cohort of 30 samples. Our results were concordant with the presence of experimentally validated and predicted monoallelic and imprinted genes. The approach reiterates the need of transcriptome sequencing as a companion diagnostic test in a genetic clinic, and these findings are instrumental to understanding the underlying pathogenic genetic lesions contributing to the pathophysiology of rare diseases. In the next step, we aim to investigate the correlation of phenotype with those genes in which the phenomenon of LOH cannot be explained by known biological reasons. Thank you.
Thank you, Numra. Okay, our next flash talk is from Afra Kaila. Is here. Yes, there. The title is the characterization of a plasmodial GDP cyclohydrolase 1 enzyme as a potential anti-malaria drug target using computational approaches. Do you need a pen drive? Is here or no? Okay, meanwhile we can continue with the next they're preparing the presentation. Sorry, the next one is Fei Hu, and the title is GACA Lite, a web server for gene set cancer analysis. Okay, there you have the screen. Follow the presentation.
Hello, everyone. This is Fei Hu, a student from Huazhong University of Science and Technology, China. I'm happy to stand here to introduce GIC Lite to you, which is a web server for gene-set cancer analysis. Since its release, it has been used by scientists around the world nearly 5,000 times. In general, after the user input a gene set, GIC Lite will analyze it, and the result can be downloaded in many formats. Our platform covered data mainly from 3 resources, including TCGA cancer multi-omic data, and drug sensitivity data, and GTAX normal expression data. This data is stored in SQLite and analyzed in our language and displayed on our web server, written in Shiny. Based on this data, we developed 8 modules for gene set analysis from multiple aspects, including MRI expression modeler, calculate the differentially expressed genes, and assesses the relationship between gene expression and the overall survival and the cancer subtypes. And the single nucleotide variation, copy number variation, and the methylation modelers explore the genomic changes and their influence on prognosis and gene expression. And the pathway activity modeler calculate the relationship between gene expression and the activity of 10 cancer-related pathways. And the drug sensitivity modeler can be used to screen biomarkers, which may influence the clinical response. We also provide a microRNA network and the GTAX expression modelers, but time is limited. If you are interested in our web server, we can discuss it during the poster time. My poster number is number 2. Thank you for your attention.
Okay. The last flush talk is the characterization of plasmodial GDP cyclohydrolase 1 enzyme as a potential antimalarial drug target. Please. Thank you, Afra.
Okay, hi everyone. My name is Afra from the research unit in Bioinformatics at Rhodes University, South Africa. So as we know, malaria remains a global health problem, and this is because of the parasite increased resistance. Sorry, I need to switch. I can't move to the next slides. Okay, I got it. Thank you for that. Right. So malaria remains a global health problem, and the reason is because of the parasite developed resistance against available anti-malaria drugs. These anti-malaria drugs, also known as anti-folate drugs, they target the parasite folate biosynthesis pathway until the resistance emerged in two enzymes in this pathway. In this study, we renew the interest in this pathway by finding an alternative enzyme, which is the first enzyme of this pathway, which is called the GTP cyclohydrolase enzyme. So to date, accurate 3D models of the enzyme of the parasite were calculated. We identified unique motifs and sequence features to ensure in future selective inhibition of this enzyme. We also identified a set of potential inhibitors from the South African Natural Compounds database. We also developed force field parameters to describe the metal centers of this enzyme. So this is the approach that we have used to characterize this enzyme. It starts with the enzyme's 3D structure. It goes both ways to the sequence analysis, in which we identify we do the phylogenetic analysis, we discover motifs, and also we perform multiple sequence alignment. It also goes to the molecular docking, in which we dock the compounds against the 3D structures, and we identify hit compounds. The force field parameters were developed using quantum mechanics calculations and potential energy surface scans. And these parameters were finally validated through the molecular dynamics simulations. We also validate the identified hit compounds through molecular dynamics simulation, and then we proposed them or regarded them as potential drug candidates for anti-malaria treatment. Thank you for listening.
Hi everyone, my name is Yvonne Gladbach, and today we move now to our industry keynote. And the industry keynote is sponsored by a platinum sponsor, Roche. And it's my great honor to introduce you to Fabian Birzele from Roche. He studied bioinformatics in Munich. He got his PhD in bioinformatics at the LMU Munich in 2009 by the group of Ralf Zimmer. He did his first postdoc at Boehringer Ingelheim about implementing NGS technologies in drug research. And in 2011, he moved to Roche in Prenzburg as a bioinformatician, and he was mostly involved in oncology projects and became a group leader for oncology bioinformatics in Basel in 2016. And now he is leading a group of, in total, 30 bioinformaticians and biostatisticians, and supporting the drug development across different disease indications from early stages to clinical trials in phase 2. Please welcome with me Fabian.
Thank you. You stole my first slide, but that's not a problem. So I'm skipping through that, okay? So that's basically me in a few minutes, in a few seconds. But maybe what I will tell you a little bit is why I went that way, okay? So just for maybe 1 minute telling you what brought me to bioinformatics. That's simple, or not so simple. I did my civil service at the time in Germany, so 1 year where I was working in a home for elder people, and I wanted to study medicine. After that, I decided, no, medicine is not my thing. And then I found the webpage from the LMU that they have a new course. In fact, this was the first semester doing bioinformatics. And I thought, well, this sounds interesting. I had math and biology as my master things. And okay, I went to that without knowing what it means. I think today it's much clearer. Then I did my PhD staying in Munich in the group of Ralf Zimmer, which is really was a very good group. Very interesting stuff there. And I worked mostly on alternative splicing and protein structure evolution. So trying to understand what's the link between splicing, what happens on the RNA level, right, and what happens to the protein structures. They need to work in the end somehow. So that was the main topic. And to be honest, we were quite limited with data. I mean, there were EST sequencing, some evidence that splicing is really happening. It was unclear how much it really happens. And then I found this postdoc position in a pharma company, Boehringer Ingelheim, where they bought one of the very first sequencing machines, I think, in industry in 2009. And that gave me the opportunity to really look in the data, in the biology itself, how much splicing is really happening. I worked there for 2 years. There was a restructuring. They didn't know if my postdoc would be converted into a permanent position. Okay, I decided to go to Penzberg, which is south of Munich, working as an oncology bioinformatician for Roche. Another restructuring brought me to Basel, so my way here was very short. I came by bike, where I took over an oncology bioinformatics team, a small team doing oncology bioinformatics. I will tell you a bit more about what we do there. And since 1.5 years, I have the honor to really lead a group of bioinformaticians and biostatisticians. We are 30 people in total. And what we do is basically the topic of that talk. And I hope it's not too boring. I'm standing between you and lunch, so I try to be not boring. And if I'm boring, you can just leave. I want to start with the main. So this is usually the last slide that Roche is presenting. And that's our purpose, if you want. And that's doing now what patients need next. And what I didn't tell you is, for me, this is the reason why I'm still working in industry and never went back to academia. Because in my current work, you really see, or we see every day, that we do something that helps drugs to move along the value chain, and hopefully we are able to treat patients who are really severely sick. So all the patients, all the drugs we develop as Roche, you don't want to take because you would be heavily sick. Cancer, immunodiseases, neurological diseases, things like that. But you really see in our daily work what your impact is, what your contribution is to that process. So Roche is a quite complex company. I just give you a brief overview that you know what this company is about. It has about 100,000 employees worldwide, and is split it into 2 main parts. There's a diagnostics part where we develop diagnostic tests for analyzing bloods or other analytes, which I think most of you, if you ever were in a hospital, there's a high chance that you were basically analyzed with a Roche diagnostics analyzing machine. And then we have a Roche Pharma part, which is also in terms of sales, the bigger part of the company. And here we try to develop drugs, okay? So we try to start very early in the development process where we identify targets, assess targets, and then we move them along the value chain into clinical phases, testing them in patients, and then also going to regulatory. Roche has, in fact, 3 really independent so-called research and early development units. You might have heard of Genentech, that's the US brand of Roche. Then there's Roche pRED, that's where I'm located in, the main, the headquarters here in Basel, and then there are multiple sites around the world, spanning from Shanghai to the US. And then there's Chugai in Japan, where is also part of the Roche family. And then once we have developed a drug to a certain level, usually clinical phase 2, which means we have shown in a patient that the drug is safe and that the drug to some extent is also efficacious, we would hand it over to another part of Roche, which is so-called PD, pharma development. And these colleagues are really responsible for developing the drug in the late clinical setting, phase 3 trials, which are then used to bring the drug to the market, to the health authorities, finally approving the drug and also producing the drug and everything that comes along with it. So my team, as I said, is about, or as you already heard, is about 30 people. And the cool thing is really that since we are in this pRED function, right, so in this department of about 2,000 people that go from very early drug development to phase 2, we really see all these projects from baby stages, from very early stages down to clinical data, which also means the analyses we do span really very early type of things down to patient samples that we see really regularly. And also we are embedded in a bigger group of about 100 people doing pathology, genetics and genomics, and proteomics. Okay, so and this is really makes it very interesting because the data generators, if you want, are really close to us. They are in the same group, we talk regularly, we meet in the coffee breaks, and we can really discuss about the technology part of it, about the protocols, which have a heavy influence on the data that we, in the end, we analyze. So it's always this back and forth that in the end makes the contribution. And since we work in a very fast-moving field, I mean, the fact that ISMBs, I don't know how many members will visit Basel today and in the next couple of days for learning more about bioinformatics. So there's a lot of new things going on. And that means innovation for us and staying on the top of science is something we have to do and which we also try to do to some extent. And for us, topics like machine learning, if you want, many people call it today advanced analytics. I personally hate that term, is something that we do regularly, and also we try to stay there. Good. So what do we do? And this is really the very high-level helicopter view of what my group is doing. If you start in the upper left corner here, in fact, we do a lot of classical bioinformatics. Sorry. Sequence homology, okay? So we just look into species and try to understand if this tox species that we want to use has a similar sequence or even has the gene that we want to analyze. Very classical things. Then we move on to a lot of mutation analyses, genetic analyses, either in big cohorts or thousands of people really doing GWAS-like things, but also somatic mutations in cancer. Then there's a bigger part on expression, right? So where we look into expression across different databases during target assessment. I will show an example in a second. We do a lot around disease relevant. Is that target, that gene really, really relevant in the context of the disease we want to treat? Here in the center, you see splicing again, which is something that more or less brought me to industry. I will show you an example in a minute. And then we, of course, do all the classical bioinformatics around interaction networks, pathway analyses, trying to understand complex expression data sets. Then we have two more, I would say, statistical topics here, which is design of experiment, trying to make sure that the experiments we conduct are really valid, which is more the work of my biostats people. And then we do a lot of biomarker data analysis. And I will show you later what a biomarker is and what we try to do there. And finally, the last slide I will show is really on something we call reverse translation, and I will explain the topic in a second. So for those of you who have never heard about drug development, this is the, let's say, blueprint of a drug development process. It's not as linear as it might seem here. And I will always highlight a little bit what bioinformaticians or bioinformatics or biostatistics does in these different steps. So the first step is to identify a target. We need to find a gene, a target, that is, if we influence that, we can basically modify the course of the disease. Okay, that's the first thing we want to do. And for that, since we have many, many target ideas in a year, last year I think my team had about 150 target assessment requests, we need to do this cheap, right? So we need to do this fast and cheap, and that's why we use in silico-based methods for that. A lot of public data that we use, a lot of internal data that we have built up over the time. And this is really used to make a case here. Is this a good target or not? As early as possible. Once you've settled for such a target, a project starts, okay? And when this project starts, you try to find a molecule, an antibody, a small chemical molecule like a kinase inhibitor, you've just heard about that, or an siRNA or whatever, to modify that gene. And in the course of finding these molecules, there's a lot of things you can do which really are driven and also helped with by bioinformatics and biostatistics. Then we really work a lot in the context of, once we have a drug, we try to understand if this drug is doing what it should. Okay, I mean, we have a hypothesis based on the target, but we don't know if we modify that target, is really something happening in a disease model, can be a mouse model, can be a cell line, can be whatever. And there you really use a lot of biostats and bioinf. And finally, it's really about in the end, clinical trials. So as I said, we are involved in up to phase 2 clinical trials. We create thousands of data points on each and every patient we see in these trials, and we also try to utilize this data then to take decisions either during the drug development or, as I will explain to you later, trying to go back to early research. The 3 examples I want to show you for today is one really toy example on target identification, one example on in vivo studies and what we do there, mostly in the context of safety, and then 2 examples more from a clinical space. So, and this is a toy example, but it's an interesting one. So, who of you has already heard about HER2? A number of you, okay. So this is a gene which in fact defines one subtype of breast cancer, HER2 positive breast cancer. And about, let's say, 20 years ago, this was more or less something which let people die very, very fast. If you had HER2 positive breast cancer, you were dead within a few months or years after diagnosis, very fast. And And then it came to the recognition that HER2 is really a driver of cancer in this particular subpopulation of patients. And it was Genentech at the time developing an antibody against that receptor. It's a receptor on the cell surface. And if you block that one, you can really treat these patients almost perfectly. They really go into remission, and many of them really survive for many, many years. If you would do this today, okay, we could just look into TCGA, the Cancer Genome Atlas. I think it was already mentioned. Now, yeah, you had a talk on more or less this data, right? We would more or less just look into that, and this is copy number data from the TCGA. So it's telling you, when it's a bit more red here, it's telling you that these patients don't have just 2 copies of the gene into the cancer genome, they have many copies of that gene in the cancer genome, which usually is an indication that, oh, something might be going on there. And since this is just a small part of the chromosome, not the whole chromosomal arm that is changing, but really just a focal, so-called focal amplification, you could rationale that there's something going on there which really means that this is an evolutionary advantage in the end for the cancer cells to have this gene amplified. Since TCGA doesn't only have copy number data, but also RNA sequencing data, you can immediately look, okay, is this gene also upregulated in cancer, right? And indeed it is. So all the patients having an amplification, if you look here, they also have indeed a very high expression of the gene. And given these two, in fact, in a very early phase of a target assessment, this would already be a good indication that we have something that is of interest to us that we could follow up on with more detailed studies. So the next step would really be to try to find an antibody or something that knocks down the gene in a cell line model and seeing if the cell lines grow less fast or whatever. And then comes the, let's say, translational aspect of it. So here you see in the middle the breast cancer cohort, right? So here's the breast cancer cohort of TCGA. Oops, sorry. But you also see that there are some outlier patients, I marked them in red for you, in other cancer indications that have very high HER2 expressions. And indeed, these are patients that might benefit from being treated with an HER2 antibody, even though it's not, it would be off-label use. It's not like we have a clinical trial showing that these patients would benefit, but it's something that might be the case. And maybe you heard about companies like Foundation Medicine and so on, so cancer sequencing in order to understand what these patients have. And if you find something like that, an amplification of HER2, you might go for an off-label use of that drug to really help the patient in the end. The second example I want to show you, we move now along the value chain from this very early baby phase type of drug to something where we try to understand safety. And this is a bit more a research project that we did with a master student that was in my group. And this is not my work, so I'm really trying to more or less not tell you anything wrong here. But it's work from David Zhang and my team who was driving that. Before we go into the topic really, just that you understand, about 25% of the drugs that are in clinical testing are not failing because of efficacy, they fail because of safety. Okay, what does it mean? It means the drug works, it does what it was intended to do, but it's not safe enough. Yeah? Patients have side effects which more or less outbalance the beneficial effect of the drug. And that's, of course, something we want to avoid. We want to know that as early as possible, that often it's just a property of the molecule. I mean, there are two options, right? Either it's a property of the molecule that makes the drug not safe because it has a metabolite that is toxic or whatever. This is something you could really try to influence during the drug development process. Or you have a so-called on-target toxicity, which means if you hit the target, if you downregulate it or modify it, this leads to simply toxicity. So that's also an option, right? In this case, it might be not the good target to go for. In the other case, it's something we can really modify and change. And we don't only want to do that, we also want to go more and more for personalized things. Again, you have to understand that at the moment, drugs are more or less giving like a one-size-fits-all pill. For example, dose, right? Your doctor will usually give you the normal dose that you are supposed to get. Don't know, if you get aspirin, you will get 500 milligrams of aspirin in one pill, right? But maybe this is the completely wrong dose for you as an individual. Maybe you would have to take twice of it because you metabolize the drug much too fast, or you metabolize it much slower, then you should only get half of it, something like that. So we try to move into more personalized spaces here to make drugs also more safe. And then, of course, it's scientifically super interesting to look into this because in the end you're moving in this area of human biology, right, where you have some in vivo testing when you move the drug to the clinic, but of course you want to be quite sure before you do that, right? You don't want to give patients something that kills the patient in the worst case. We have rat models or mouse models that we can use for that, which we can either do in vivo or in vitro. That's something we can do easier than human testing. And we have human in vitro data. And this more or less quadruple or 4 things we can look into is something we really try to understand better. So there's one data set, which is a public data set, TG Gates, where people have really done that. They have looked into 170 compounds. They have really done a very good study in terms of control and different doses for these. They have looked into primary hepatocytes, primary rat hepatocytes, and then they have looked into rat liver and rat kidney. Why liver and kidney? Because these are the 2 metabolizing or main metabolizing organs in human, right? So drugs usually are either metabolized in the liver or are secreted through the kidney. And if you see a drug side effect of a small molecule, it usually happens or often happens in the liver or the kidney first. So this is a big data set, and we have here, so the important bit for the story I will tell in the next 2 slides is that we have here pathology data. So we have pathologists looking at these kidneys and livers of these rats, trying to understand what has happened there, what did the drug do to liver and kidney, and we have expression profiles. And the idea David came up with, together with Tao Fang, this master student that we had in the team, was, okay, if we take the expression data, can we somehow predict what the pathologists would have found in the pathology data? That was the idea. And they did that using different machine learning methods and also some what is today called deep neural networks. And you can see that indeed, so the green bars here always tell you that the deep neural network indeed outperforms the other different classifiers. And these are more or less the different type of pathology readouts. What's that? Okay. The different pathology readouts that you get. And you also see that different pathology readouts are more difficult or easier to see in the corresponding pathology expression data, which is clear. Some of them give a very clear expression signal to know if something goes to necrosis. That might be easier to see than if something goes to something else. The last example I want to show you is really about biomarkers. So first of all, what is a biomarker? A biomarker is something we can measure in the best case, a patient, or at least in an animal, that we can measure from blood or from other tissue biopsies, whatever it is, trying to understand what the drug does in the body of the patient. Or trying to understand if the patient has a very bad disease or a very good disease. So HER2, for example, as I said, HER2 positive breast cancer was measured based on biopsies from the patient. It would be something that was a so-called prognostic marker because patients who had the disease died very fast in the course of the disease. That's the first thing, prognostic power. So you can really take the marker, you say, well, this patient is super sick or this patient is not so sick. The patient will likely be okay again, or this patient, in the worst case, might die. Then there's biomarkers which are more diagnostic. So we have a big bunch of patients, right? We measure the biomarker, whatever it is, trying to see, okay, in this case, I give drug X, and in the other case, I give drug Y, which is more the personalized type of thing we want to go for. And finally, and that's the thing we always are looking for most, or which is the holy grail of biomarkers in drug development, that is the question, can I say at baseline for a patient, if I give this drug, the patient will be healthy or good again or not? Yeah? Why are we interested so much in that? Simply because we don't want to treat a very severely sick patient, like a cancer patient who maybe has 1 or 2 chances of treatment with something wrong, right? We want to give this patient something which really has a high probability of helping this person. And this is called a predictive or biomarker. So the story I will tell you is about a program we had, which in the end didn't work out, so the program was stopped. And this was on a target called CD44. CD44 is a very complex gene, so it has many different known isoforms. You can only see here some of them. For the story I will tell you, the important one is this here, which is the CD44S variant. So all these variable exons here are skipped in this splice variant. What did I do? Okay. Can I just close it? Yes. And the others were these variable exons here that you see here. Is there a pointer, by the way? Yeah. Wonderful. So here you can see these variable exons that can be included in the gene or not. Okay. And we had a program around that, CD44, that you can find thousands of publications implicating this gene in cancer. So saying this is a relevant cancer target, you should block it, and so on and so forth. And we had an antibody against that protein, which was really binding to all different isoforms. It was binding somewhere here. So it bound all the isoforms. However, we had some, okay, now I cannot move on, interesting. Yeah. We had some xenograph data, and xenograph is really mouse models, or mice that were, where you have a human cancer growing in these mice, okay? And we had about 40 of these xenographs, so a pretty extensive data set with 40 different human cancer cell lines. And when we, and this was done without knowing what the isoforms are, without even having this data set. When I joined Roche, so about 2011, we had the chance to do RNA sequencing on all the cancer cell lines we were using as a standard in our programs. And also we did these 40 cell lines here. And when we had the data, suddenly we found something very interesting. We found that out of the cell lines that have CD4S expressed, so this isoform here, 50% of these cell lines were responding to treatment, which is not too bad. But from the CD44V expressing cell lines, so these here, exactly 0 cell lines were responding to treatment. So the chance that you have a responder if CD44V is the main isoform being expressed was exactly 0. And we tried to, so this meant that we might have found a predictive biomarker for treatment. At least it was a sufficient but not necessary condition that the cell lines would express CD44S. We did some follow-up experiments on this. We did try to link it to biology. So CD44 is a molecule that binds hyaluronic acid. That's, by the way, something people eat to have their skin being more elastic. And it binds, and indeed what we see is that CD44S cell lines express much more of this hyaluronic acid than CD44V cell lines, and there were some other biological conditions that we looked into and understood a bit better. And then our biomarker people came and said, okay, well, that's all nice, but does it mean anything for the clinical development of the drug, right? It's xenograph findings. Do we even have this difference in expression in human population, human cell lines, and that in human cells? And at the time, we had the chance to look into TCGA, which just came out, and we found that. So indeed, the expression of CD44V, that's the red bars here, versus CD44S, which is the blue bars, is very, very different in different cancer indications. So, give an example, AML. All AML patients express CD44S. More or less none of them expresses CD44V. Colon cancer, few patients express CD44S. More or less all of them express CD44V. And to remember you, CD44V is the variant we don't want because we are quite sure the drug will not work, at least based on our xenograph data. Okay, when we showed that, our dear clinical colleagues got a shock because the molecule was already in clinic, but it was in colorectal cancer, so here. And that meant, according to our data, the likelihood that the drug would work in this indication was almost 0, because most of the patients would have had CD44V. And then we really tried to turn the ship around. We tried to inform the investigator. So these very early studies are all-comer studies, right? So more or less all patients where the investigator says we can try, it's maybe the last resort to treat with that antibody, would be eligible for the treatment. So we tried to turn the ship around. We tried to really find patients which were falling into this CD44S cancers. We tried to enrich for different cancers which had CD44S. There was no test that we could use other than RNA sequencing, which was too slow at the time. And yeah, in the end, the molecule didn't work. But for the few patients where we had data on, and we really saw that if there was any response at all, and to be very fair here, response is very, very low. Okay, so this is a so-called RECIST plot. I think the colleague before from Novartis was showing RECIST as well. So what it means is if it's below 0, it means the tumor shrinks. Okay? If it's above that, it means the tumor grows in the course of treatment. And we can say that the only patients who had some kind of a signal indeed were expressing CD45S, by the way, despite being colorectal cancer patients. So somehow our story seems to hold true. Nevertheless, the molecule was stopped after several years of investigation and research simply because the response rates were not as they were expected and were not good enough. This happens. Good. So what will keep us busy for the next couple of months, years to come? That's something which we call, at least in Roche, forward and reverse translation. So traditionally, what we as bioinformaticians do since many, many years and what drug development is like is we go from research using public data, using internal data to the clinic. We try to support clinical development as much as possible in a process that we now call forward translation. In the end, we want to find the right drug for the right patients. We want to understand and manage the side effects, and we want to use our data to optimize the development of the drug. Now, what we see in the last couple of years is there's a huge growth in the availability of clinical data. And the source for that is twofold. First of all, we as Roche also in our clinical trials generate much more high-dimensional data. We do a lot of RNA sequencing on the patients. We do a lot of genome sequencing on the patients. We measure many other additional type of things depending on the study. And there's a lot of other data, like you might have heard about Flatiron, which is a company doing electronic health record mining, or more or less doing electronic health records. And there are many others, like Patients and Me. There's IBM Watson around. There's a company that Roche bought. By the way, both of these companies were just recently bought by Roche Foundation Medicine as well. And we try to use this data to do something different, namely to go from clinic, which is in the end the population we want to look at. This is that's where the information has to come from, right? Not from some animal models. We want to go the way back to research in order to understand our diseases that we want to treat better, in order to identify novel targets, and in order to develop more personalized drugs. And that's something that will keep us busy because the complexity that comes with that and comes with all the clinical data that we have access to are really huge. And that's also where I think we as a team are super well positioned because we have the bioinformatics, which is important for all the high-dimensional data, and also the biostatistics with us, which is the people that really, really understand how to deal with such a clinical trial and how to deal with the complex data that comes with it. So, conclusions. I think bioinformatics and biostatistics today are really core elements of drug development. And we really help our colleagues to drive decisions along the whole value chain. I've shown you some examples, and there are many more. I personally liked that job of a research oncology bioinformatician always very much because it's extremely diverse. I mean, the data you get is always top-notch. You get a lot of data, and you have always the newest data types. You're currently working a lot with single-cell sequencing data, for example, and also try to bring that to the clinic, right? Bring that to the patients. And in the end, I mean, making sense of all that is a challenge, but that's also where the fun comes from. If you are someone who's close to his PhD or a postdoc, and if you are an expert in analyzing metagenomic data, you can write me an email because we are looking for one colleague who could do this for us. Okay. Other than that, we are hiring sometimes. You can just find everything on the web. But you can also, if you are really interested to hear more, write me an email. And we also have usually interns in the group. We have usually postdocs, 1 or 2, and we also have master students if you are still at the level of a master. Good. That's it. Back to the purpose. And if you have questions, let me know. Can you understood most of it, but not everything?
For a clinical trial that's already underway, like your bioinformatics study comes up with a potential issue that suggests this trial cannot possibly work. Yeah. Do you stop the trial? Do you trust it?
So I mean, in this case of CD44, they were not stopping the trial. They were just trying to amend the protocol, right? To go for disease indications, in this case, tumor indications that where the likelihood was higher that the drug would work. But you know, I mean, if your evidence is extremely strong, let's say you would find a very, very strong safety signal that tells you, well, this drug is super cancerogenius and you should not really give it, then you likely would have you would very sure have an effect on it, right? If it's more fluffy, you have to discuss and convince the clinical team in the end that they should change something. That's a good question. Yeah? To think like repurposing a drug that's already on the market? Yeah. So we do that, yes. But it's, you know, I mean, we don't look for public drugs which are somehow done by someone else. I mean, we do this for our drugs and try to see if we can do any label extension or any extension of the thing. To be very honest, I think this is usually at the moment something that's mostly done by the late stage colleagues. So they really, I mean, they follow a drug and they have the antibody in their hands, and then they really do also maybe some trials together with investigators to see if the drug works in another indication. For us, it would be more like, would we find the same pathway or the same gene to be involved in other diseases where we didn't think it would be the case? And that would be, I think, a classical case where you try to take that and try to move back. I don't have any example where this already worked, but I'm pretty sure there must be a lot out there that you could do. You know, I mean, I think you won't bring down costs by that, because in the end, you would have to run another trial to show that the drug works in this other indication. I mean, what you save is the development of the drug. And then you have to be honest, then you have to think about patent life, right? Because the antibody that's already on the market has a certain patent life, and then you have to think very carefully how much patent life you have left and how much you can do about it. Yeah, this one first, and then you.
Can you comment on the use of in silico methods for like lead generation? I know there's like a lot of noise about like structure-based drug discovery and docking and stuff like that. Is that like just noise because hard throughput experiments is just easier, or is those sort of in silico methods used in practice?
You mean for finding the right molecules?
Yeah.
I'm not the expert there, so my team is not doing that. We have a team of a few more or less chem informaticians, right, that really look into this. I'm sure they do that, but I honestly don't know how much it's done. I know that we also do antibody modeling, for example, through really looking into crystal structures and trying to modify wherever possible, but I'm not the right person. Difficult question, and I don't have the answer. So Foundation Medicine is doing genomic testing, right? So this is really trying to for cancer patients to provide a test that tells them you have these mutations, and usually it's a targeted test. It's about 500 genes that they look into. And yes, you see these patients, just like I've shown in one of my first slides, that have a HER2 amplification in an indication that is not expected, right? But I have no clue about the percentages of that. Regarding real-world data, to be very honest, at the moment, real-world data like you have in Flatiron is super messy, right? I mean, you can do many interesting things with it, but the data, even though it's a relatively high standard of curation, it's still not, I mean, you don't have any high-dimensional data, for example, right? What we have now is we have one data set where we have the combination of Flatiron patients who have been measured by a Foundation Medicine test. So we have about a few thousand patients where we have both, and there you can do a bit more, because then you can start to correlate, survival times, for example, with mutation profiles. That's very interesting, but I'm personally waiting for RNA sequencing data to come into Flatiron data. Then, for me, for my team, this would be where the game is really changing. Yeah. Yeah, I mean, Roche is investing heavily in this personalized healthcare field, right? We have our own department group doing only that. You would have to ask these people how fast they think they will really make a difference in the drug development process. I think it's maybe 5 years ahead, right? And honestly, I think the examples that will come will be more like these repurposing type of examples, not so much the ones where you really, I mean, a drug project takes 10 years, right? So we will not see drugs on the market in the next, unless timelines for clinical development are much faster, maybe due to real-world data, because you don't maybe need a control arm anymore. You can do this from a synthetic control arm, right? But I don't know. It's still some years ahead.
I wonder if you can comment on why you're interested in studying genomics. Are you thinking that that's forward translation direction or the reverse translation direction? Are there clinical settings actually?
Yeah, so I personally think it's more the reverse one. Yeah, so we are really planning to do to embed metagenomics in more of our clinical trials. And there are some really very concrete plans where also the person that would get this job would really work on that. And yeah, I see it more on the reverse part. Any other questions? All ready for lunch? Good, then happy lunch.
Well, this is the end of the first session. Some announcements. Please, the speaker of the next session, before some minutes start, the second session can bring us the presentation, please. Now we have the lunch. I think the lunch should be there outside. I can see the bags from here. So just go out and take the bags. That is the lunch. Well, and where's the poster station? Okay, that's the poster station will be in the first floor. If you have some time now, you can hand your posters. Oh, well, thank you very much for your attendance to the first session.